Hello, everyone. I'm Tiff Bortwood, the Managing Director of Australian Genomics. Welcome to DNA Dialogue, the webinar series where we share new developments in health genomics and hear from international leaders in the field. As we come together today, let's take a moment to acknowledge the traditional custodians of all the lands we're joining from today, the lands of Aboriginal and Torres Strait Islander people. I'm joining you from Naarm, country of the Boonwurrung people of the Kulin Nation. I pay my respects to elders past and present and extend that respect to all First Nations peoples joining us today. So it's my very great pleasure today to introduce Marc Fiume, entrepreneur, researcher, and global opinion leader. Marc is focused on realising the future of precision healthcare with standards and technologies that help scientists leverage the collective power of the world's exponentially growing volumes of omics and health data. Marc is the founder and CEO at DNAstack and has an active role in GA4GH, the Global Alliance for Genomics and Health. So Marc, today, we're all very well aware of the problems of siloed data sets from different institutions, clouds, devices, countries, jurisdictions. And today, Marc, we're presenting a vision for accelerating collaborative research by connecting and analysing distributed data sets and federated networks. Marc, thank you so much for joining us today.
Thank you so much for having us.
Oh, it's wonderful to have you. So please use the Q&A function throughout the presentation, Marc's presentation. I'll get to as many questions as I can at the end, and we'll pepper Marc accordingly with all your questions and comments. Also, if you're active on social media, we encourage you to use the hashtag DNA Dialogue, and please tag at @OzGenomics. So Marc, I will hand over to you, and I look forward to having a chat at the end of your talk.
Wonderful. Thank you so much, Tiffany, and everyone for the opportunity to share our vision for creating a federated future, one where we can all benefit from working collaboratively towards facing some of the biggest challenges in health. So here's the agenda today. I want to tell you more about how we see the opportunity to talk about the principle of federation, why that's different from where we've been with data centralization, to share our vision for the future, to talk about standards that we've been developing along with the Global Alliance for Genomics and Health for doing so, to talk about a platform called Omics AI that makes it easier for us to create these sort of internet for genomics and health research, tools that we've been using to make discoveries, and finally to round out about talking about some of the networks that we've been using together with our partners to find causes of rare and common disorders. So first off, I want to tell you a little bit about my son. This is Alexander. He's turning 8 years old this year. Last year, Alexander was diagnosed with epilepsy. He was 6.5 years old, and we had just gotten through the pandemic, and all of a sudden he had a cluster of seizures, and 3 of them came in the span of a week's time, this time not febrile. So we visited the neurologist who confirmed his diagnosis through a sleep-deprived EEG. And as a father and a parent of a child who's recently been diagnosed, you get flooded with questions about your child's disorder. What kind of epilepsy is it? What's the prognosis? What are the triggers? What are the causes? And if so, are they genetic or something else? Would he be able to swim? Can he drive when he gets older? So all of these questions come to mind, and somewhat frustratingly, not many of them have answers. And what I found in my experience and what I've learned is actually the common experience of others who are going through the epilepsy journey, is that treatment is very often an experiment of trial and error, where you're prescribed a medication, and if you have symptoms or side effects, you take a new medication and you iterate, sometimes over the course of 30 years, in order to find the one that's right for you. And in a world where So much of our lives is digital. Increasingly, our health data is becoming digital. We must be able to learn from each other's experiences to improve healthcare outcomes for all of us. When we look at one of the biggest problems that prevents us from getting there, it's that health data are siloed. They're siloed by device, by database, by organization, by province, by state, by country, by cloud. And so while data represent about 30% of all of data, we're really not optimizing its use. So the question becomes, how then can we unlock its potential across these silos? So at DNAstack, and this is a mission we share with many other organizations as part of the Global Alliance for Genomics and Health, it's to accelerate life-saving discoveries through the creation of technology that enables us to harness the collective power of the world's omics and health data. And in our mind, the future is federated. What we mean by that is that we want to move away from the old way of sharing data, where data custodians generate high-value data sets, and in order to co-analyze them, they have to upload copies across the internet to a central location or database in order to ask questions of it. The future we want to enable is a new way of sharing insights, where instead of moving data to the questions, we move questions to the data and only communicate the answer back. So with federation, we can make discoveries across distributed data sets really critically without moving them. And this increases data volumes and diversity, while at the exact same time enhancing privacy and security. It's a win-win. So our vision is to help enable a future where precision medicine is powered by a real-time, federated, global sort of internet for omics and health research. And to do that, we know we need standards. So you can go and build a network that's proprietary, but if you look at the most successful federated networks, things like the internet and telecommunications, they're built upon a foundation of open standards. So for the internet, we have HTTP and TCP/IP, SMTP. For telecommunications, we have 5G, SMS, and GPS. In a much similar way, we have the Global Alliance for Genomics and Health. That's this international standards body comprised of 500 organizations or more from around the world who are all working together to create technical, ethical, regulatory, and security standards for omics and health data. We're really careful about building standards for real-world problems, and that's why we've architected the GA4GH to develop technologies and standards that are aligned with the problem statements and pain points articulated by real-world driver projects. So you'll see this image is sort of a matrix diagram where the rows of the matrix represent technical work streams. These are the functional kind of working units of the GA4GH. And the columns represent real-world driver projects. These are driver projects that articulate their needs. And the marriage between them represent where the work happens. There are 29 real-world driver projects, including all of us, EDI, Genomics England, IPCHIP, and really importantly, Australian Genomics represents a wonderful driver project from which we've learned so much from. So at DNAstack, we wanted to create technology that, much like the regular internet, makes it easy for people to connect, access, and analyze omics and health data across them. So Omics AI is a product suite that we launched in September of last year, actually the last plenary meeting of the GA4GH. And it's comprised of 3 interoperable modules. The first one is called Publisher, and that's for data custodians to be able to connect, organize, protect, and share data across these global networks. The second one is called Explore, which is, if you're thinking about this as a federated network, it's the hub of the network that enables researchers to be able to easily find, access, and query data sets. And finally, we have Workbench, which allows for secondary and tertiary analyses across the universe of data that's connected to those networks. We've been really careful not to lock customers and researchers into specific environments. And so we've made sure to develop the platform so that it's interoperable with multiple instruments, multiple clouds, and and people can use these software in regions according to international privacy regulations. So taken together, Federation and Omics AI really unlock a number of new benefits. So because data reside in your own environment, it increases your sovereignty. Because you control who has access to your data and for what purposes, it increases security. Because you avoid storing and transferring many copies of very large data sets, it's more cost efficient. Because you decide what specific questions people can ask of your data, it's more privacy preserving than sharing row level information. Because it's very easy to add new nodes to these federated networks, it's more scalable. And because you're not locked into a particular instrument or cloud or region or vendor, it enables more freedom. So Omics AI is fully compliant with a number of GA4GH standards, from data representation, data discovery, data access, data analysis, and more. And again, it helps our partners because of all these features. It helps them comply with international privacy laws, which sort of govern the movement and custodianship of personal health information. So I'm going to really briefly switch over to a browser window to show you how some of these products work. Okay, so this is Publisher, and this is for data custodians to be able to govern their data and share it over GA4GH standards into federated networks. So I'm going to refresh my page here. And so the first step for the data custodian's journey is to connect a data source. And we've built data source connectors to popular object stores like AWS S3, Google Cloud Storage, Microsoft Azure Blob Storage. And in the event that you operate on premise, we have MinIO. The idea is that whether your data is represented as a file or a table, we can connect to it. But really importantly, we don't take copies. We create an index. So once you've connected any number of data sources, those data sources are indexed into your library. So here you have sort of a master index of all of the data that's been connected across my data sources. And the next step is to organize those data into what we call collections. It's kind of like creating a Spotify playlist. So here is one collection that we worked together with Genome Canada as part of Canada's national genomic surveillance efforts through the COVID-19 pandemic. So on a nightly basis, we would receive raw viral genomics data. We would run a standardized secondary analysis pipeline, and we would redistribute the results to researchers, government agencies, pharmaceutical companies, anyone who wanted to consume the data sort of in real time as Canada was monitoring the circulating viruses that it was collecting as part of its testing framework. So a collection is described with a name and a rich text description. And in the description, you could link out to, say, documentation, acknowledgement, terms of use, etc. But sort of the most important aspect of the publisher is we wanted to make it very easy for data custodians to become GA4GH compliant. So this is sort of the GA4GHification interface, where with a click of a few buttons, you could enable GA4GH APIs on top of your data. So the first API I'll show you is called search. And by enabling search, you become compliant with GA4GH Data Connect standard. And by enabling download, you become compliant with the GA4GH Data Repository Service standard. And for each one of these services, you can choose to make your data available publicly, registered, which means you have to have an account, or controlled, which means you have to have an account, and you need to be added to an allow list. Once you've activated GA4GH APIs, you can enable discovery, which makes it registered to any number of federated networks. And for the purpose of this viral genomics sequence database, we've connected it to a network that we've created called Viral AI. So that's the data custodian's experience in creating one node in a federated network. So I'm going to switch over to an instance of the second piece of software that we described, which is called Explorer. Again, Explorer is a federated network of data that's been published to it by any number of data custodians. Really importantly, each one of these cards represents a distinct collection that may be physically and administratively controlled by a number of different data custodians. And again, these data can be stored in any cloud in any region. It is truly a federated network, but from the perspective of the researcher, it appears as if it's one experience. So by contrast to the Canadian data set, I could look at the NCBI SRA SARS-CoV-2 genomes cohort, and I have, with the exact same representation, samples, variants, and files that were curated from the NCBI. So this, for the first time, by virtue of the data being standardized to the same GA4GH specification, enables us to seamlessly co-analyze data across federated networks as if it were sitting on my laptop. So if you can click into any one of those cards, you have this sort of interactive interface for doing faceted search across the collection of data. And we, as I'll show in a minute, we have a number of different tools that allow you to do more sophisticated analysis if you wanted to do, say, a downstream statistical analysis, visualization, or machine learning experiment. The last product we have, it's called Workbench. And Workbench is a piece of software that allows you to run statistical analysis, sort of secondary, tertiary analytics on computational environments that are local to the data. So really excitingly, we would be able to add an engine in any one of these clouds as listed here, in AWS, in Google Cloud, Microsoft Azure, or on-prem through a connector to the workflow execution service, and in any one of the regions that they support. So really it's multi-cloud, multi-region, with the intent of enabling data to be computed on where it lives. Once you've connected one or more engines, you're able to select a workflow, and you may have seen interfaces for this before. Essentially I can pick a workflow, inspect the versions, run the latest version, specify inputs to the workflow, and at runtime, prescribe the specific environment that and region that I want to run this workflow in. So that's Workbench. I'm going to flip back now to my presentation, and I'm going to talk about other tools that we can use to drive discoveries. So by virtue of the fact that we've GA4-GHified a large and growing network of data, sometimes in specialty-specific disease areas. I talked about infectious disease being one area of focus for the last 3 years, but we've also been standing up other networks in, say, rare disease and in neuroscience and soon to be oncology. But by virtue of all of these data sets being represented in a common open standard, it's created an open innovation ecosystem where new applications can be built on top of the network. I'm going to show you a few applications that we've been developing in order to extract value from the data, but the whole intent for us GA4-GHifying this data is to enable a new kind of opportunity for data scientists and developers to be able to easily create new applications. So one application that we're working on, it's called Questions. And the idea behind Questions is instead of sharing row-level information about the contents of your database, you instead provide data custodians a way to prescribe very specific privacy-preserving questions, like what's the allele frequency of a specific genetic variant? Or in the context of a gene, what's the mutational burden across individuals in your database? A specific version of a question that we've worked on for many years is called Beacon. And Beacon is a sort of genomic go fish. It's a way for one organization to identify other organizations who've seen patients with similar genotypes and soon to be phenotypes in the latest version of the specification. So you enter in a genetic variant of interest, and it scours this federated network for other organizations who have seen the same thing. We have a Google Sheets integration, which enables the ability to search across our networks, not within the interface, but using a function call. And this is the way for our partners to be able to compose really custom visual analytics dashboards for themselves. We have a Python client, which enables you to consume the data within Jupyter Notebooks. That's an environment for interactive visualization data analysis. As I mentioned, we have Workbench for running non-interactive secondary and tertiary analysis pipelines. And we've really worked hard with some of our partners, including Integrate AI and NVIDIA Flare, to create toolkits for conducting federated learning across networks, that is, training machine learning models across federated networks without moving data outside of any one of the nodes that we've created. Finally, we are working on an application called Conversations. It's inspired by the growth in conversational AI and large language models, which is an AI-powered tool to be able to answer the questions that you have across the network that are posed in natural language, whether those are posed in English, French, or some other language. This, we think, is a very powerful new interface that's going to make data science much more accessible to non-technical users. Finally, I want to talk about networks. These are networks that we're growing together with partners and are eager to continue to partner to scale out the impact that they're having in diverse areas of research. So we are working with some incredible partners, advocacy groups like Autism Speaks, hospitals like Sick Kids here in Toronto, sequencing manufacturers like PacBio, pharmaceutical companies like Roche, government agencies, including the Department of Research and National Defense in Canada, and some international research consortia like IPCHIP. So one of those consortia is called Aligning Science Across Parkinson's. It is a collaborative research network that will be announced early in the summer, working to create a network of research organizations, all publishing data to accelerate a path to a cure in Parkinson's. That's led by our fantastic partners at the Michael J. Fox Foundation, together with a number of different research teams. The second one, which is a great connection we have to Australia, is IPCHIP. It's the International Precision Child Health Partnership, creating a network across the Hospital for Sick Children's, Boston Children's, Murdoch Children's Research Institute, and UCL in London. And that is also a driver project, one of the 29 for the Global Alliance for Genomics and Health. I thought one of these testimonials from a researcher was really telling. This is Dr. Evdokia Anagnostiu. She's the VP of Research at Holland Bloor View Kids Rehab Hospital. And we work with Evdokia and our team to create a federated learning model across 5 pediatric hospitals to predict medication use from demographic information. And she said that the ability to explore multiple data sets from different jurisdictions will give us both the volume and diversity of data to have the power to make transformative discoveries for the lives of kids, youth, and adults with developmental differences and disabilities. Crucially, doing so without having to move the data from local environments will enable us to respect the requests of various communities to protect their privacy and confidentiality. So I thought that was a really wonderful kind of quote from a great partner of ours, and we're really excited to continue to working with like-minded partners to drive new impacts and discoveries. In summary, we've shared our perspective on the opportunity that lies ahead of us to learn from each other's health experiences. We believe that because health data is large, sensitive, and regulated, that the future is federated. Our vision is to create a global federated internet for omics and health research that's built upon the principles and standards of the Global Alliance for Genomics and Health. Omics AI, as introduced, is a new software platform launched last year that is making it easy for us to support our partners in creating GA4GH compliant federated networks. And this has created a sort of open innovation ecosystem for applications to drive new discoveries. And just really excited and grateful for the opportunity to work with some incredible partners to create new networks. So innovation, I'll say, is absolutely a team sport. DNAstack has had the pleasure of working with experts in ethics, cloud computing, genome science, genome sequencing, patient advocacy, and more. And we're really, really grateful for a whole ecosystem of partners that have aligned with this vision. So with that, I would just like to close and say thank you very much. and maybe advertise that we're excited to come visit you all in Melbourne, Australia later this year to kind of celebrate the 12th plenary meeting of the Global Alliance for Genomics and Health. So with that, thank you very much to Australian Genomics and TIFF for hosting us. Thanks.
Mark, thank you. That was just an excellent presentation. And, certainly your parallels to Go Fish and making a Spotify playlist, etc., makes it a very accessible presentation also. Now, I have to apologise to everybody. We're having a bit of an issue with the Q&A in that I can't see any questions. And we know we've posted a couple of test questions. So apologies to those of you who've asked lots of questions in the chat. But we have got some questions on our local team chat. So Miranda asks about the structure where analysis is done, where the data is located. She asked, does that mean the cost of the analysis is then the burden of the data custodian for cloud? Or if it is an on-prem instance, are local HPCs managing the compute for external queries? And if so, how are users managing this cost or compute burden?
A really wonderful question. I'll say that the cost for the questions feature, which I mentioned, which is something like an SQL query that's packaged and parameterized, that is, essentially free. It's an SQL query that runs. It's actually low cost. That typically is the cost is borne at the level of the data custodian. Where more significant costs can be incurred is if you wanted to do a statistical analysis that's much more computationally intensive. For that use case, it is possible to co-locate an engine in the researcher's tenancy in the cloud and region where the data reside. There is data movement between them, although you don't incur, say, significant egress fees by virtue of it doesn't have to leave the region or cloud that the data are located in. So the longer answer is the short answer to that, to my long answer is yes, it's possible, and we can configure it both for researchers to pay or the data custodian to pay.
Okay, that's clear. Thank you. Catherine just texted me a question. What do you see as the current gaps in the use of DNAstack to federate and analyze raw genomic data?
Good question. I think that there's two. I would say most importantly on the policy and legal side, we're getting accustomed to changing our vernacular from sharing data to sharing insights. And in our experience with Beacon, sharing a Beacon style question gets us through privacy and security challenges much more than sharing row level data about our patients. So I think one is about how we change our vernacular to start talking about federated insights. And then the second challenge we have is on data harmonization. So typically, we leave that as out of scope, and we get our collaborators in a room to say, hey, come to a consensus on how you harmonize our data. That's not the most elegant solution, and often we have mismatches to how people process their data. So that is quite often a sticking point, is if you want to do a federated analysis across two data sets that are not represented in the same way, there can be a gap there.
All right. Excellent. Thank you, Mark. So MJ asked a very good question. Often data, particularly healthcare data, is a bit of a mess in the data sets. How have you found working with clients to get the data in formats where it can be put into federated networks and queried?
Linking to the answer to the last question, we've only been able to solve that by working together with partners. And in fact, we've built out a data science and bioinformatics team to service the data hygiene, cleanliness, standardization of the data as it gets put into the system. So we do have, alongside the products, a team of bioinformaticians and data scientists to support that effort.
All right. I don't envy their work, I tell you that.
They're busy.
So Louise thanks you for the presentation and asked whether you're incorporating care standards to protect data from Indigenous and other underserviced and represented groups.
We are actively working with that sort of conversation in those groups within Canada as well. The feedback so far has been really, really good in terms of empowering underrepresented communities to be the custodians of their own data instead of having to rely on a trusted third party. So that has gone a long way with building trust and empowering Indigenous communities to kind of participate on their own terms in these networks.
Mm. We have in prior webinars had presentations from Crystal Soti and Rodney Herring, and so it's certainly a topic that we've explored in these series before. So I think it's certainly a lot of opportunity to engage with and ensure that the needs of Indigenous data sovereignty are met with DNAstack's products. More text. This is great. Thanks, people, for texting questions. Hamish asked a silly question in his words. How many different open standard tools are there, and are they interchangeable? So how do they compare with commercial tools, for example?
Oh, that's a good question. Tools, I showed about a half dozen of them. There are about a half dozen more which I didn't show, which people are actively using. Platforms like Terra is an example,
Yeah.
non-DNAstack platform through which you can consume the data. That's our thesis has never been to sort of lock in analysis into one environment. The idea is to let there be a proliferation of tools to be able to take data from the platform and out. So if you're a developer, if you're a data scientist, we would continue to love your feedback in terms of what environments are the most exciting for you to be able to analyze your data in. Recently we've got a lot of requests for, say, TensorFlow, these kind of like AI machine learning environments. So we're always open to new and creative ideas for how we can make the data as impactful as needed.
Excellent. All right. So people will be getting in touch with you then if they've got other platforms. Yeah. So Kerry noted that you mentioned that patient advocacy groups are partners. So Kerry is progressing and managing our project on Involve Australia, which has developed guidelines for the involvement of the community in research and also sort of policy areas in health. So she's interested in what impact these patient advocacy partners have had and how have they been involved in DNAstack projects.
We love working with patient advocacy groups. We feel like our thesis is so aligned in that they are sort of the closest representation of the patient as we can get without speaking directly to the patient. So for us, both in terms of their kind of posture on open science and impact and wanting to share the data as broadly as possible, that's been really great alignment and vision. But also we're using that as an opportunity to become more engaged in the cohorts and populations we aim to serve. So one of the initiatives we led was called the Autism Sharing Initiative, which aimed to create the first global federated network of genomics and health data in autism. And through that process, we worked with Autism Speaks, who as a proxy to the voice of the autistic community worldwide, was really helpful in providing a lot of helpful feedback in terms of what would be the most beneficial thing to build in service to their needs. And so we use advocacy groups as sort of the voice of the customer from the perspective of the patient and the individual. And it's really, really been a driving force for how we develop the products.
Mm. I would imagine that, particularly in the instance of complex conditions like autism, the leveraging these tools to be able to query and interrogate across large data sets would be of particular value. Yeah. So the lovely Di Nichols emailed me a question. Please send me questions in any format you'd like. So this is Diane Nicol from Ruse. What a great talk. As a parent of a sick child yourself, what are the big concerns do you think parents and patients might have in allowing their data to be accessible in this way? And what strategies do you have to explain the complexities of genomics, AI, et cetera, to them? Which is a lovely sort of linking question to your last response.
Good question. Of course, you're always so the first concern is how can I get the best care for my son or daughter? And that for me trumps a lot of the other concerns, which is I just want to make sure that, as we give a medication to my son that affects his brain chemistry, how is it working and is it the right thing for him?
Mm.
As a secondary concern, of course, I would say, so the first thing I did actually when we got the diagnosis was I contacted a bunch of my genomics friends to say, hey, how can I get Alexander into a study? Because we'd love to contribute to the research. As part of contributing his data to research, I would be, of course, concerned about its use for, discrimination. And so we always want to be mindful of unintended uses of data. But that for me is sort of a secondary concern. And in terms of how I communicate to him, was the question framed how I communicate to my son
Oh,
around the
not
use
to
of the
your
data?
son. Let me just open that up. Um,
So as you
how do
do that,
you, yeah, more to, yeah, parents and patients. So, data donors, I guess.
so I wanted to go through this experience myself. So I registered for the Personal Genomics Project Canada,
Mm-hmm.
and the idea behind PGP Canada, and PGP, there are actually satellite efforts going in different countries, but the idea is to do whole genome sequencing, um, a loose kind of clinical workup, and to share the data publicly as an exercise on the willingness of individuals to collect data and share genomics data with the research community. And that for me was an eye-opening experience to just go through it for myself.
Mm.
And of course, the first concern is, do I have anything that um, would significantly change the course of my treatment or my perspective on the future? And luckily, when I received those results and went through genetic counseling, there was nothing really. I was in a position that I didn't feel I could share openly. And I think that kind of exercise, kind of thinking through with empathy how a patient or an individual would feel, really helps you think about how you as a custodian of that data should be protecting of it. But yeah, in our experience and going through it personally with my son, again, it goes back to, I think most individuals are very giving and trusting of their data, for the purpose of really helping others with similar conditions going through the same journey.
That's a really thoughtful answer. Thank you. I think there's many on the call are sort of involved in data governance or data custodians, and I think it's a lovely principle that we can bring to the processes for sharing our data. Matilda asks, with respect to consent, we have vast differences on how and where data can be shared. So you mentioned public versus private access approved. What granularity exists to refine or restrict sharing within data sets? I mean, this is,
Yes.
despite the
Yeah.
efforts of Ruse and Dury and GA4GH, there is a lot of variation in consent.
Yes. So we leave the consent collection process up to the custodian, but we provide the tools to be able to share the data following the consent. The knobs you can turn are which services your data are exposed to. Right now we have search, download, and questions, and those have sort of varying degrees of privacy. And then on top of each one of those services, you can choose whether the service is open to anonymous users of the internet, which is public, registered users, which means you have to have an account, but you don't go through a data access committee review. And then finally, controlled access, which is you have to go through a data access committee review and be approved. So both in terms of what services are enabled, and who can access those services. We provide you the ability to tune those to the consent.
All right, excellent. Catherine asked another question. Can you speak to the capacity to share clinical data and potentially linking to electronic medical records?
Yes, so we don't connect directly to medical records. What we have typically done is to lift data from a medical record into a research database and then connect it through the publisher that you've seen. So the shorter answer is yes, we do it, but we do it in a two-step process as opposed to a direct connection.
All right, excellent. Matilda asks, so she's interested in DNAstack's evaluation and impact measurements. So in terms of winning over ethics approvers, policymakers, and governments to adopt such data sharing practices and tools, there's certainly metrics as to the number of lives impacted, and this sort of evaluation metrics would be really valuable.
Yeah, that's a really important question and a two-part answer. So I totally agree with the premise, and the work that we've done so far has provided some visibility for data custodians to be able to communicate to their funders, sometimes philanthropists or government agencies, what have you, on the increase in impact of their data in terms of the consumption, the number of users, etc. What we'd like to implement as a stage 2 is something more sophisticated, right? As we're running federated analyses where you might make a discovery that's a composite of sort of multiple databases, how does each data custodian receive appropriate attribution relative to the contribution to the total discovery? And so as a technological solution, we are going to be implementing something called value attribution, which captures the relative contribution your data set has made to a discovery that was asked in a federated context. So we hope that's a sort of a new incentive system and a new way to measure your impact that you can then communicate to your funders.
I think that that approach would be of great interest to the Australian Research Data Commons. So they've been putting in place processes to increase the value of research data sets, particularly clinical trial data sets, with secondary use and access to those data sets. But that was a key concern of those who would be making their data sets available to others. How do they get recognition and attribution of their contribution? Yeah.
We feel like that could be a new metric alongside the number of citations, is how impactful was your data and how impactful was the data that you shared into a network. Yeah.
I think too, it's with examples, back to Matilda's question about, measuring this impact. I think too, examples like the IPCHIP collaboration where it's up and running, it's clearly leading to benefit not only academically but clinically for patients involved. I think it's helpful in the context of Australian genomics advocacy for moving towards federated approaches to genomic information management, having exemplars like DNAstack and Omics AI to be able to say, look, it is achievable, it is feasible, look, it's up and running. I think that's a really sort of valuable from our messaging as well, because it does seem almost when we speak to policymakers about it, it's almost put in the too hard basket.
Yeah, we're super grateful to the leadership at Murdoch and the other partners in IPCHIP. We think it's going to be a world-leading initiative that will hopefully generate discoveries in a new way.
So we've exhausted, or have we exhausted the questions? Yes, we have exhausted the questions, but people will have an opportunity to ask further questions and have more of a chat with you at the 12th plenary in Melbourne in September. So I'd really encourage everybody to register. The registration is now open. We're very much looking forward to welcoming Marc and oh no, John's just come through with another question. Here's John. Are you considering the possibility of integrating other omic data sets? And if so, what additional challenges do you see?
Yes, so we are actively integrating metabolomics, proteomics. The platform is agnostic to data type, so we can support anything that could be represented as a file or a table, and all the same challenges still apply. So I would say data harmonization continues to be the challenge, but it's not anything different than we're already tackling with DNA. Great question.
Oh God, they're coming thick and fast. So Emma asks, great presentation and vision. Oh, you've touched on this a little earlier. So how does costs for the software work? Like, what does it cost clients? I suppose you can't go into that in too much detail. How does it get funded? So long-term viability of these sort of networks and the products. So the time and resources for a client to adopt the product.
Yeah, so in terms of time, the biggest component of the time is the legal sign-off for sharing data. Once we get clearance to deploy, it can be deployed in under a week. So we could actually move very, very quickly. Our business model is to license the components of our software. So there's a fee associated with the publisher and the explorer. Workbench we provide for free. And we are working really hard to decrease our own costs around publisher and explorer to really minimize the costs to the data custodians. Most of our data custodians are research organizations, advocacy groups, for which funding is constantly a challenge. And so it really is our goal to democratize access to the infrastructure. So when faced with the consideration to build versus buy, that is just a really cost-efficient option for them. So I'd love, if you have more questions around costing, I'd be very happy to answer them. But yeah, that's where we are.
No, I think that's certainly an excellent approach. Caitlin asks, are there any specific challenges or considerations? So, adding to the topic of consent, are there specific challenges or consideration if a patient decides to withdraw their consent for sharing?
Yes, yeah. So we have the right to be forgotten. We have procedures around how that happens. Typically, we can remove data completely from the system in about 10 days. So, yeah.
That's amazing. That's very responsive. All right. Thank you, everybody, for your flexibility in asking questions through various alternate channels rather than the Q&A function. And Marc, thank you so much for joining us today. It was a really fascinating presentation and lovely to have a chat with you. And we really look forward to seeing you in Melbourne in September.
Thank you so much for the invitation. Yeah, really looking forward to seeing you all there.
Excellent. And thank you all for joining Marc and I today for this presentation. Our next DNA Dialogue seminar will be on Thursday, the 2nd of May, and will feature Mary Herbert, Professor of Reproductive Biology at Newcastle University from the UK. So please join me then. Thanks again.